While people all over the world have been struck by its stunning imprint of the face of Jesus Christ, not everyone's convinced that the shroud is a genuine holy relic.
In 2015, within a highly sterile genetics lab at the University of Padua, Professor Gianni Barcaccia and his researchers at last unraveled the genetic material concealed in the Turin Shroud, the fabric that countless Christians think covered Jesus Christ's body following his crucifixion. From the moment the shroud truly entered historical records, there has been debate surrounding it, along with doubts about whether it's genuine. They anticipated something straightforward, a primary genetic profile, either from Europe if it was a fake from the Middle Ages, or from the Middle East if it was real. Instead, they discovered something else entirely. The genetic material didn't link to a single individual, one nation, or even one region. It connected to China, to India, to East Africa, to old groups so remote that their DNA remained unchanged for millennia. The secret sequence embedded in those threads outlined a path so extensive and exact that it couldn't have been added deliberately, couldn't have been counterfeited, and couldn't be accounted for by any idea that believers or doubters had ever suggested. This is how they uncovered it, and why it alters everything. The face nobody was meant to witness. To grasp why that genetic material demolished every prior explanation, we have to begin with the evening when the shroud first disclosed what it concealed. May 28, 1898. Turin, Italy. A lawyer and hobbyist photographer named Secondo Pia received special approval from King Umberto I to capture images of the artifact during a public showing. Taking pictures was tough labor in those days, slow, precise, relentless. Pia carried a camera almost as big as a trunk up onto platforms inside the cathedral, employing magnesium bursts and strong lights to illuminate the shadowy space. He developed 2 big glass plates, each 50 by 60 centimeters in size. Later that night, by himself in his darkroom, illuminated solely by the faint shine of a red safety light, Pia dipped one plate into the developing solution. As the picture emerged, his hands began trembling. He almost let the glass slip. What was appearing in front of him wasn't supposed to be there. On the photographic negative, where bright areas should turn dark and dark ones bright, a clear, high-contrast face materialized. Sunken eyes softly shut. A fractured nose, a mustache, and divided beard, swelling on the right side of the face. For the first time ever, the form concealed in the cloth wasn't a vague outline. It was unmistakably a man. Serene, noble, but showing signs of intense agony. The impact was profound. This wasn't merely an unexpected find. It was a pivotal moment that entirely shifted how the shroud was perceived. Here's the twist. Any ordinary picture acts in a foreseeable way as a negative. Bright becomes dark, dark becomes bright. And the outcome seems warped and artificial. But the shroud violated every norm. When photographed as a negative, it looked like an actual photo of a genuine person. What the bare eye observes on the material is already a negative. Who in the Middle Ages grasped photography 800 years prior to the invention of the camera? Who could produce a perfect negative image without any means to view, experiment, or verify the outcome? Nobody. The human eye can't perceive the world in negative form, much less recreate it with accurate mastery over brightness and shade. Photography wouldn't come into existence for another 800 years. No painter, no researcher, no expert living then had the understanding to even imagine such a concept, let alone carry it out perfectly on a section of linen. This was the initial breach in the barrier of doubt. The shroud didn't act like an artwork. It acted like a photographic surface seizing a lone moment in time, and that insight caused a ripple through every field that would eventually examine this fabric. If this exploration is already drawing you deeper, a like and a subscription are the easiest methods to assist us in continuing to reveal tales like this. Your backing truly counts. But that photo was only the prelude. The true revelation was lurking in the particles, and when they finally interpreted it, even the doubters fell quiet. The DNA that shouldn't be there. For over 600 years, the argument about the shroud stayed stuck. Belief on one side, reason on the other, neither ready to yield. For those with faith, it was the sole tangible leftover connected to the resurrection, a mute observer to the occurrence that transformed history. For doubters, it was the most sophisticated fake from the Middle Ages ever made, perhaps created by Leonardo da Vinci personally, or some unidentified expert meant to trick visitors seeking wonders. But in the 21st century, guesswork yielded to something tougher to dispute. Genetics, high-energy physics, spectroscopy, forensic molecular examination. Experts ceased viewing the shroud as a sacred item and began handling it like a crime site, a biological information storage, silently safeguarding 2,000 years of the past. In 2015, Professor Barcaccia's group of genetic experts and biologists obtained unmatched entry to the artifact. Their aim wasn't to locate the DNA of divinity. Science lacks a framework for what such evidence would resemble. There's no baseline example, no benchmark for evaluation. They sought to rebuild the fabric's own history, where it had journeyed, who had contacted it, whose grasp had clutched it through the ages. Employing clean micro-vacuum tools fitted with extremely fine sieves, they carefully gathered tiny dust, pollen, and natural remnants from far within the weave and woof strands, areas where old substances can stay protected for thousands of years. They also studied bits gathered during the 1988 repair. This effort stretched science to its boundaries. Even the smallest current pollution, a breath, a snort, a sneeze, a lone skin particle could erase ancient indicators and substitute them with genetic interference. And here's the key. They concentrated especially on mitochondrial DNA, and for an essential purpose. Unlike nuclear DNA, mitochondrial DNA is present in hundreds of duplicates per cell. It's inherited solely via the mother's side. It endures much longer in old substances, and it acts as a strong, dependable indicator of regional source, group shifts, and human relocation. If any genetic data had persisted within those threads over 2,000 years, this was the approach that would detect it. For weeks, machines operated without pause, interpreting millions of nucleotide patterns and matching them against worldwide genomic collections depicting groups from across the globe. When the ultimate charts showed up on the display, haplogroups, family lines, regional beginnings, the lab became silent. No one uttered a word. Barcaccia's team was gazing at information that contradicted every forecast they had formed. This was not the genetic mark of one person. It was something much more intricate, something nobody had foreseen. The discoveries, released in the esteemed journal Scientific Reports, exploded like a detonation throughout the scientific world. The investigators had predicted a distinct primary genetic indicator. If the shroud was a fake from the Middle Ages crafted in France, as doubters have long asserted, European DNA, particularly French or Italian, should have heavily prevailed in the findings. If it was a genuine artifact from Jerusalem that never departed the area, Middle Eastern DNA should have prevailed. Neither occurred. What surfaced was a whole planet inscribed on one fabric. First, the Middle East. Haplogroups often linked to the Druze, a closely connected, historically secluded society in Israel, Jordan, Lebanon, and Syria, whose genetics have scarcely altered in thousands of years, a potent sign of regional source offering solid proof of a Middle Eastern link. Second, Western Europe haplogroups U5b and H, 1 through H3, aligning with hundreds of years of European contact by the Poor Clare nuns who mended it, the House of Savoy who possessed it, and innumerable visitors who honored it across the ages. Third, and this is where it becomes astonishing, North and East Africa, haplogroup L3 directing toward Egypt and Ethiopia. This was both unforeseen and profoundly intriguing, indicating direct interaction with some of the oldest Christian groups on the African continent. Fourth, South Asia, haplogroups M39, M56, and R8, genetic signs usual in the Indian subcontinent, and most astounding of all, East Asia, haplogroups D4 and G2. Typically connected with China, India, Africa, Europe, the Middle East, all inscribed into one old fabric. But how? And if a counterfeiter didn't place it there, who did? The solution was concealed in an old pathway that most experts had already rejected as legend, a planet inscribed on fabric. Now, focus on this. No craftsman from the Middle Ages laboring in a French monastery around 1350 could have assembled genetic substances from China, India, and East Africa. In the world of the Middle Ages, globalization wasn't a thing. Long-distance journeys did happen, but not on a level large enough to deposit such broad, plainly recognizable genetic marks on one piece of material. These were societies known only via far-off travelers' accounts like those of Marco Polo. Nor could such an individual have deliberately inserted those remnants on a fabric in a manner that would deceive genetic researchers 600 years afterward. The DNA discovered on the shroud is not haphazard pollution. It is a shared biological recollection. But when Barcaccia plotted the data on a map, the enigma grew even more profound. The spread of genetic indicators matched with striking accuracy along the Mendelian route. The ancient trail experts had long discarded as myth. Based on early Byzantine, Syrian, and Arabic records, the shroud was folded so solely the face was seen, a style termed the tetradiplon, and shown within a unique frame. This artifact didn't remain in one location, and it definitely didn't originate in Europe. Its travels began in Jerusalem. From there, it shifted to Edessa in the 2nd century, where the shroud stayed concealed inside the city fortifications for hundreds of years. And here's the key point. Edessa was positioned at one of the most vital intersections of the ancient world, the Great Silk Road. Caravans from China, India, Persia, and Arabia traveled through the city. Traders transporting silk and spices, pilgrims and envoys from remote areas. All arrived to revere the image thought to guard the city. They stood nearby. They kissed its enclosure. They contacted the fabric. And with each meeting, tiny remnants, skin cells, hair pieces, drops of perspiration settled onto the material. Layer upon layer, century upon century, the DNA of the planet built up on its surface like unseen dust. The travels didn't conclude there. From Edessa to Constantinople in 944 AD, the largest megacity of the ancient era, where individuals of every ethnicity and background gathered. Then in 1204, during the Fourth Crusade, the city was savagely plundered. The shroud vanished. It re-emerged in Athens around 1205, passed through the possession of French knights, and ultimately surfaced in France around 1353 in the keeping of Knight Geoffroy de Charny. And it is here that the genetic proof strikes a crushing hit to the theory of a fake from the Middle Ages. The DNA on the shroud is not arbitrary disturbance. It is a locational imprint that no counterfeiter could have produced. Dust from footwear, garments, and hands left by innumerable individuals who stood in front of this face over many hundreds of years. But the genetics were merely part of the concealed sequence. The remaining part was inscribed in flora, and it directed right to a crime location: pollen, blood, and a crown of thorns. Professor Avinoam Danon, a distinguished Israeli botanist from the Hebrew University of Jerusalem, and Swiss forensic expert Max Frey, separately analyzed pollen grains captured deep inside the linen threads. When Danon viewed the full outcomes, he recognized the consequences were huge. 58 distinct plant types pinpointed, 17 indigenous to Europe, anticipated considering 6 centuries of recorded European contact, but the bulk originated from other places: the Middle East, Turkey, and the Anatolian steppe, flawlessly aligning with the old path through Edessa and Constantinople. This is where it becomes extraordinary. Scientists pinpointed plants that flourish nowhere else on the planet, only inside a slim path between Jerusalem and Jericho. One type prevailed, Gundelia tournefortii, a spiny desert thistle. Its pollen constituted almost half of all specimens, focused intensely around the head and shoulders. Why would thorn pollen drench a burial fabric? The crown of thorns. Gundelia tournefortii, with its extended needle-like points, is precisely the plant Roman troops could have woven into a derisive crown. It flowers near Jerusalem in early spring, exactly during Passover. A second plant hint came from Zygophyllum dumosum, a species limited to a very confined area, growing solely in the Judean desert and sections of the Sinai Peninsula. Its pollen was also discovered in notable amounts. No counterfeiter from the Middle Ages operating in France could have acquired pollen from plants native only to Israel, much less distributed it invisibly at a microscopic scale over a piece of fabric. A counterfeiter would have employed pigment. Pollen can't be applied like paint. It serves as an unseen stamp from a crime location, a locational imprint that can't be counterfeited. And the blood narrated its own harsh tale. For numerous years, doubters contended that the reddish marks on the shroud were simply colors: ochre, cinnabar, or tempera blended with gelatin. That assertion fell apart in 2017. A group headed by Professor Giulio Fanti at the University of Padua, collaborating with doctors from a hospital in Trieste, inspected the marks using transmission electron microscopy and Raman spectroscopy. At the nanoscale, they observed not color, but blood. The blood located on the material was confirmed as human and of blood type AB, one of the scarcest blood groups globally. This identical blood type shows up repeatedly on old Christian artifacts, including the Sudarium of Oviedo. But here's the key point. This was not the blood of a fit individual. Inside it, researchers identified nanoparticles of creatinine and ferritin attached to hemoglobin. Such intense levels happen under solely one group of circumstances: catastrophic fatal injury, extended torment mixed with dehydration and extensive muscle breakdown. When muscle tissue is continually harmed, a condition called rhabdomyolysis, creatinine surges into the bloodstream in massive quantities, and the blood conserved on the fabric documented precisely that. This is not emblematic. It is a biochemical cry of suffering. The man enveloped in this fabric was assaulted to a bodily condition already unable to sustain life before crucifixion even started. Proof of more than 100 strikes matching Roman scourging using the flagrum. Leather lashes inserted with lead balls, and the blood remained red, which should be unfeasible for old blood. Usually aged blood darkens to brown or black, but scientific review disclosed abnormally elevated levels of bilirubin, a material discharged by the liver during intense pressure and serious injury. Bilirubin maintains the red hue of blood for hundreds of years. The blood of a tormented man remains red, not mysticism. Biochemistry under intolerable strain. A painter can mimic the look of a lesion. No human grasp can duplicate the biochemical mark of intense injury, organ breakdown, and hypovolemic shock. Every particle directed to the identical location and the identical occurrence. But what about the single test that was intended to demonstrate it was all a counterfeit? The test that erred. In 1988, 3 renowned labs, Oxford, Zurich, and Arizona, carbon-14 dated the material to between 1260 and 1390 with 95% assurance. Medieval, a fake. Matter resolved. The globe embraced the judgment. Even the Catholic Church withdrew, calling the shroud an icon instead of an authentic relic. But wait, because science doesn't halt. 3 decades afterward, investigators found where the vital error had occurred. And the issue wasn't the tech. It was the human choice of where to extract the specimen. In 1988, a small segment of material, no bigger than a stamp, was sliced from the very border of the shroud. That edge had been managed innumerable times across the centuries, taking in sweat, skin oils, candle wax, and microbes. Then, chemist Ray Rogers of Los Alamos National Laboratory uncovered something ruinous. That edge had been skillfully mended during the Middle Ages. The Poor Clare nuns who restored the shroud strengthened the impaired border by interlacing new cotton strands into the original material, colored with alizarin and secured with gum arabic to blend with the aged linen. The primary section of the shroud has no cotton whatsoever. It is composed completely of linen. The labs didn't date the shroud, they dated the repair. In 2022, physicist Liberato De Caro at the Institute of Crystallography in Bari presented a fundamentally distinct method: wide-angle X-ray scattering, or WAXS. Rather than examining pollutants, this technique assesses the aging of the linen cellulose itself at the atomic scale. Over time, linen naturally deteriorates. The polymer chains of cellulose gradually fragment under contact with radiation, moisture, and heat. This mechanism acts as the substance's inner timer. De Caro contrasted specimens from the shroud with materials of established age, from Egyptian mummy coverings going back to 3000 BC to textiles from the Middle Ages. When the aging graph showed on his screen, De Caro summoned his associates right away. The shroud's atomic arrangement didn't align with fabrics from the Middle Ages at all. It was much more ancient. It aligned with linen retrieved from the stronghold of Masada in Israel. Dated between 55 and 74 AD 1st century, the era of Christ. In 1988, science rejected the wonder. In 2022, it restored it. But one enigma still looms over everything else, and current physics can't clarify it. The unachievable image. No strokes from a brush, no colors, no dye. The image is present only on the outermost 200 nanometers of the threads, hundreds of times slimmer than a human hair. It is a chemical alteration triggered by oxidation and dehydration, a sort of burn mark left by an unidentified energy origin. Researchers at ANEA, Italy's National Agency for New Technologies, Energy and Sustainable Economic Development, attempted all possibilities to replicate it. Acids, warmth, gamma rays. Nothing succeeded. Only a short, powerful surge of vacuum ultraviolet radiation approached it. Tech that didn't exist in the old world. To embed the image over almost 4 square meters of material, the body would have needed to emit a flash of energy enduring less than a billionth of a second. Strong enough to imprint the fabric, yet exact enough not to incinerate it. And that's not even the most insane aspect. In 1976, NASA examination showed the image holds flawlessly precise three-dimensional data. The strength of the mark matches precisely to the gap between the body and the fabric. This is something no creator, old or current, has ever managed to copy. Digital Review also spotted forms over the eyes aligning with coins resembling uncommon lepta struck under Pontius Pilate around 29 AD. The chance that a forger from the Middle Ages could have known or recreated this element is basically nil. The nails go through the wrists, not the hands, precisely as forensic research foresees for crucifixion. Even nerve reactions are evident, including the pulling back of the thumbs due to nail harm to the median nerve. Layer upon layer, biology, chemistry, physics, geology, numismatics, everything points to Jerusalem between 30 and 33 AD. Here is what nobody can evade. A forger from the Middle Ages would have required to counterfeit a photographic negative hundreds of years before photography. Insert pollen from plants located only between Jerusalem and Jericho. Create blood with injury indicators undiscovered for 600 years. Copy three-dimensional encoding no contemporary facility can reproduce. And gather DNA from China, India, Africa, and the Middle East. All from a French monastery with a candle and a paintbrush. The shroud is not a painting. It is a forensic account of a real human being. A biological repository and a mute observer to an instant that changed human history. The concealed DNA sequence plotted across its threads follows a 2,000-year path that no forger in history could have composed. So the query is, do you think science will someday clarify what occurred inside that tomb? Or are some occurrences past the grasp of any tool we'll ever construct? Share your response in the comments. And if this shifted how you view the shroud, subscribe so you don't miss what we uncover.